All right, so get this. We're diving into the world of microbes today.
Oh, microbes. Exciting.
Yeah. Specifically, we're going to unravel the story of Aspergillus oryzae.
Aspergillus oryzae.
You might know it as koji.
Koji, okay.
This mold, it's basically royalty in Asian kitchens.
Really? Like how so?
It's the powerhouse behind soy sauce, sake, miso.
Oh, wow, I didn't realize.
And a whole bunch of other deliciousness.
Sounds like a pretty important mold.
But here's the thing, this deep dive gets really interesting.
Okay, I'm listening.
Because Aspergillus oryzae has this sort of like evil twin.
An evil twin. What do you mean?
It's called Aspergillus flavus, lurking in its family tree.
Oh, interesting. Closely related but different.
Exactly. This close relative, it produces aflatoxins.
Aflatoxins, those are...
Some seriously nasty toxins.
Yeah, they contaminate crops.
Right.
And they can cause some real health risks worldwide.
So the question is, how did one mold become a culinary hero?
Right.
While the other is a global health concern?
So like two sides of the same coin, kind of?
Yeah, kind of. Get ready to explore microbial domestication.
Microbial domestication. I'm intrigued.
We're going to uncover the surprising journey of how a potentially dangerous mold became a kitchen staple.
I'm ready. Let's do it.
So I got to say, it's fascinating how appearances can be so deceiving with these molds.
Oh, really? How so?
They look almost identical.
You're kidding.
Yet their impacts couldn't be more different.
Wow.
Aspergillus oryzae, take its aroma, for example.
Okay, the aroma.
It's kind of like a mix of mushroom, grapefruit, and like a touch of sourness.
Interesting.
It's master at breaking down starches and proteins.
So that's how it gets those complex, savory flavors.
Exactly, the flavors we love in fermented foods.
Right.
But then you have Aspergillus flavus.
Yes, the evil twin.
It's less about enhancing flavors.
And more about, well, producing those harmful toxins.
Right, okay, so we've got this Jekyll and Hyde situation going on.
Jekyll and Hyde, that's a good way to put it.
In the mold world. But how does microbial domestication fit into this?
Good question.
Sounds like humans might have had a hand in shaping Aspergillus oryzae into the culinary superstar it is today.
Yeah, so just like humans domesticated wolves into dogs,
right,
we've kind of been guiding the evolution of Aspergillus oryzae.
Over thousands of years.
Thousands of years, exactly.
Oh.
Early fermenters, maybe without even knowing it. They selected for traits that made it better at fermenting.
Interesting.
And crucially, they reduced its toxicity.
So it was a gradual process.
Yeah, but there's a catch. Unlike, domesticated animals, microbial domestication, it often happens at a metabolic level.
Meaning?
It's harder to spot those evolutionary shifts. They're hidden.
So it's not like we can just look at Aspergillus oryzae.
Right, exactly.
And see how it's changed over time.
Exactly. These changes are happening deep down in its genes.
Wow, that's wild.
One of the biggest changes, it's a genetic deletion.
Oh, a deletion. What does that mean?
It means a part of its genetic code is missing.
Huh.
And this deletion, it disables Aspergillus oryzae's ability to produce aflatoxins.
Wait, so it lost the ability to make those toxins?
It did.
That doesn't sound very advantageous.
You'd think so, right? Losing a defense mechanism.
Yeah.
But here's the thing, aflatoxins, they're actually toxic to yeast.
Really? To yeast?
Yeah, and yeast is crucial for fermentation.
Oh, wow, I didn't know that.
So, by ditching the toxin production, Aspergillus oryzae accidentally made a better environment for yeast to thrive.
So by giving up its toxins, it became a better fermenter?
Exactly. It formed a better partnership with the yeast.
Fascinating. But there's more to it than just ditching the toxins, right?
You got it. This mold also became a starch-digesting powerhouse.
Yeah, that's what I was thinking. How did that happen?
It's a prime example of what we call an energy trade-off.
Energy trade-off, okay.
So without the burden of producing toxins, Aspergillus oryzae could suddenly redirect its energy to breaking down starches and sugars more efficiently.
Interesting. So it's like it's specialized.
Exactly. And the evidence is right there in its genes.
What do you mean?
Scientists, they've discovered Aspergillus oryzae has multiple copies of this gene called the alpha amylase gene.
Alpha amylase, what does that do?
It produces an enzyme key for breaking down starch.
So it's like it doubled down on its starch digesting abilities.
You got it. And humans, they unknowingly selected for these super efficient starch digesters.
Wow, that's incredible. But how long did this domestication process take?
That's where it gets really surprising. Microbial domestication can be much faster than you might think.
Really? Like how fast?
Let's take Penicillium camemberti, the mold responsible for camembert and brie cheeses.
Oh yeah, delicious.
So it likely evolved from its wild relative Penicillium commune, which has a less appealing, kind of musty odor.
Right, not so good for cheese.
Right, but in a lab, when they grew Penicillium commune on cheese, for just a few weeks.
Just a few weeks, wow.
It started to change. Lost its pigment, stopped producing toxins.
Really?
Even lost that musty odor. And you know what it developed?
What?
Those buttery, cheesy aromas we love in Camembert.
So that's a wild mold transforming into a cheese-making superstar.
Exactly.
In a matter of weeks. That's incredibly fast.
It is. And it all comes down to the environment.
The environment. Okay, explain that.
The environment plays a massive role in how microbes evolve.
Okay.
So for Penicillium camemberti, that cheese environment...
Lots of fats and proteins?
Exactly. It became this selective pressure.
It favored the molds that could thrive in those conditions.
You got it. The ones that couldn't adapt.
They didn't survive to pass on their genes.
Exactly. And this intense pressure, it speeds up evolution.
Like a fast-forward button on evolution.
Exactly. So thinking about Aspergillus oryzae's journey to domestication,
yeah.
Did its environment play a similar role in its transformation?
That's a great question. Let's think about that. We need to go way back in time.
Way back.
Perhaps even to our early hominid ancestors.
Wow, that's going way back.
And their relationship with fermented foods.
Interesting.
Imagine early humans encountering fruit that had naturally fermented.
Okay, I can imagine that.
Due to wild yeasts. You know, they might have noticed the taste change.
A change for the better, hopefully.
And maybe even some mind-altering effects.
Who knows?
So are we talking about the very origins of our love for fermented foods?
Maybe. It's a possibility.
And Aspergillus oryzae, with its love for rice.
Right, its affinity for rice.
It might have been part of the story from the beginning.
How so?
Imagine early humans gathering wild rice. Those grains could have easily had Aspergillus oryzae spores.
Oh, interesting.
And then, as humans started cultivating rice, it created a perfect opportunity for this mold to flourish.
It's like a symbiotic relationship developing.
So from accidental encounters on wild rice to becoming a staple in cultivated rice.
Wow.
The relationship between humans and Aspergillus oryzae deepened over time.
That's a great point.
This seems like a good place to pause for now.
Okay.
I'm already thinking about all the ways early humans might have figured out how to harness the power of Aspergillus oryzae.
That takes us to the Neolithic Revolution, a pivotal moment in human history. And for Aspergillus oryzae, too, but we'll have to save that fascinating chapter for next time.
So we're back, still diving deep into Aspergillus oryzae.
Back into the mold world.
We left off talking about those early humans and their first encounters with this amazing mold.
Right, those early encounters.
Now we're heading into the Neolithic Revolution.
A big turning point for humans.
Totally. And as we'll see, it took our relationship with Aspergillus oryzae to a whole new level.
I bet. I mean, think about it.
Yeah.
The Neolithic Revolution, it completely reshaped human societies.
For sure.
As our ancestors transitioned from hunter-gatherers to settled farmers.
Right, big change.
They suddenly had this challenge, this new problem.
What was that?
Preserving all that excess food.
Oh, that's right. They had more food than they could eat right away.
And that's where fermentation really comes into its own.
Makes sense. Fermentation helps preserve food.
Exactly. Take cheese, for example.
Okay, cheese.
Fresh milk, it spoils so quickly.
Yeah, it does.
But a hard cheese, that can last for months, even years.
Wow, that's amazing.
So you can see how fermentation became this essential tool.
Oh, yeah.
Especially for those early civilizations.
So it wasn't just about making food taste different.
No, not at all.
It was about making it last longer, a crucial technology.
It was a game changer for those early farming communities.
Totally. But did they know back then, did they have any idea that Aspergillus oryzae was at work?
Well, there's some pretty compelling evidence that suggests the intentional use of Aspergillus in fermentation, going back thousands of years.
Thousands? Really?
Yeah. For example, in China, there's a site called Jiahu, and they found pottery shards there dating back 9,000 years.
9,000 years.
Imagine that. And these shards, they contain residues.
Residues of?
A fermented beverage.
Oh, interesting. Like what?
Made from rice, honey, and fruit. Think prehistoric rice wine.
Wow, that's fascinating.
So it seems like our ancestors, they were deliberately using Aspergillus.
Way back then.
Long before they understood what it even was.
That's incredible. We've been talking about how Aspergillus oryzae works with yeast during fermentation.
Yeah, that partnership.
What was that like back then? Was it like a finely tuned partnership like we see today?
That's a good question. It's important to remember that early fermentation, it was largely a matter of chance and observation.
Oh, interesting. So they didn't really know what was happening.
Right. They relied on what we call spontaneous fermentation.
Spontaneous fermentation. What's that?
Basically, they would notice that certain foods, when left in specific conditions, they would, undergo these natural transformations.
I see. So it was about observing patterns.
Exactly. And then trying to recreate those conditions.
To get the same results.
Exactly. So you can imagine those early fermenters.
Yeah.
They might observe that, say, rice, left in a certain environment,
Okay.
it would transform into this flavorful,
Oh, interesting.
maybe slightly alcoholic beverage.
So that's spontaneous fermentation, just letting nature do its thing.
Right. But eventually, humans, they started taking a more active role.
Okay, how so?
They wanted to make fermentation more predictable.
Right, more consistent.
Exactly. And they developed these really ingenious methods.
Like what?
One of the cleverest was backslopping.
Backslopping, what is that?
Think of it like sourdough starter.
Oh, okay.
You take a bit of a successful fermentation.
Okay, filled with those good microbes.
Exactly. And you add it to a fresh batch of ingredients.
I see. So you're ensuring that the good microbes are there from the start.
Precisely. This made the process way more reliable.
Makes sense.
And much more controlled.
So they were cultivating the microbes they wanted.
In a way, yeah.
Without even knowing exactly what they were.
It's pretty remarkable when you think about it.
It is. When do we start to see evidence of intentional Aspergillus oryzae cultivation?
Well, there are ancient Chinese texts.
Okay.
Dating back to 300 BCE.
Wow, that's old.
And they specifically mention using Aspergillus oryzae.
So they knew about it by then?
They did. They even had techniques.
What kind of techniques?
For breeding the mold on steamed rice.
Interesting.
Carefully collecting and drying its spores.
Oh, wow.
All for controlled fermentation.
So that's a big shift, right?
Absolutely. From spontaneous fermentation to the deliberate cultivation of Aspergillus oryzae.
That's a huge step in the history of fermentation.
It really is.
It's amazing how humans, through observation and experimentation, went from relying on chance encounters.
Yeah, to mastering, the art of microbial cultivation.
It's incredible. But I'm curious, as techniques became more sophisticated, did they start to understand that different strains of Aspergillus oryzae produced different results?
Absolutely. Think about different dog breeds.
Okay, dog breeds.
Yeah, different breeds with specific traits, temperaments.
Right, right.
Well, there are also diverse strains of Aspergillus oryzae.
I see, like different versions of the mold.
Exactly, each with its own unique characteristics.
That affect the fermentation process.
Right. Some strains might be better at breaking down certain starches.
While others create different aromas or flavors.
You got it. And skilled fermenters.
Okay.
Guided by generations of, tradition and intuition, they learned to select and cultivate the ideal strains.
Fascinating. So they were picking the right mold for the job.
Exactly. Depending on what they were making, a delicate steak or a robust soy sauce.
Wow, that's amazing. It's like this dance.
Yeah, delicate dance.
Between science and tradition.
Exactly.
So they weren't just cultivating Aspergillus oryzae. They were cultivating specific versions of it.
To get the exact flavors and textures they wanted in their fermented foods.
That's incredible. It really highlights something important about microbial domestication.
What's that?
It's not just about taming those wild microbes.
Right.
It's about actively shaping their evolution.
To meet our needs, our desires.
Exactly. Humans become like microbial architects.
I like that. Microbial architects.
Designing and shaping these microbial communities.
To create the foods and beverages we enjoy.
And that brings up a question for me. We've talked about the benefits.
Yeah.
But are there any downsides to domesticating Aspergillus oryzae?
That's a great question, and it's an important one.
Yeah.
Because, you see, as we focus on cultivating those specific strains...
The ones with the desirable traits?
Exactly. We might unintentionally be narrowing the genetic pool.
Oh, I see what you mean. Less diversity.
Right. And that loss of diversity, it could make those domesticated strains more vulnerable
to what?
To diseases, or maybe less adaptable to environmental changes.
So it's like putting all our eggs in one basket.
It is, kind of. By focusing on just a few select strains, we might be losing valuable genetic resources.
That could be important down the line.
Exactly. For future adaptations, for resilience.
It's like the importance of biodiversity in nature.
Yes.
But on a microbial scale.
Precisely. So as we continue to refine our fermentation practices
and explore the potential of Aspergillus oryzae,
we need to think carefully about how to balance the benefits of domestication
with the importance of preserving microbial biodiversity.
Exactly. That's a crucial point.
Well, as we wrap up this part of our deep dive, I'm really struck by the journey we've taken.
Yeah, it's been quite a journey.
From those chance encounters with wild Aspergillus oryzae to the deliberate cultivation of specific strains.
It's a story that shows the ingenuity and adaptability
of both humans and microbes.
Exactly.
It really is.
And it highlights just how powerful this ancient partnership
between humans and microbes really is.
But there's still so much more to explore.
There is.
Oh, yeah. In the last part of our deep dive, we're going to delve into the modern applications of Aspergillus oryzae. And we'll explore its potential, its amazing potential.
Exciting.
To address some of the challenges facing our world today.
All right, welcome back to the final part of our Aspergillus oryzae deep dive.
Back for more mold talk.
You know it. We've explored its history, its domestication, even its role in, shaping human evolution.
It's been quite a journey.
It has. But this mold, it's more than just a culinary hero.
Is that right?
It's got some surprising talents that go way beyond the kitchen.
Like what? I'm intrigued.
Well, what's fascinating is how its talents are being applied in ways that could totally revolutionize different industries.
Really? Give me some examples.
For instance, its powerful enzymes.
Enzymes, okay.
They're being used in biotechnology.
Oh, interesting.
To develop, new pharmaceuticals.
Pharmaceuticals, wow.
To create biofuels.
Biofuels, too.
Even to produce those eco-friendly detergents.
No kidding. So we're going from, soy sauce and sake to, medicine and biofuels.
That's exactly it. It's quite a jump.
It is. How does a mold that, breaks down starches in food end up being so useful in all these different fields?
It all comes down to those amazing enzymes it produces.
Right, the enzymes.
Remember how we were talking about its ability to break down those complex starches and proteins? Well, those enzymes, they're like tiny molecular machines.
Molecular machines, okay.
They can perform very specific tasks.
Like what kinds of tasks?
Scientists have figured out how to use them for a whole bunch of applications.
Okay, like what?
For instance, they can break down plant materials for biofuel production. Or they can create new types of polymers.
Interesting. Polymers, okay.
Yeah, for those biodegradable plastics.
Wow, that's pretty cool. So Aspergillus oryzae is basically giving us this toolbox of enzymes.
That's a great way to put it.
That we can use to, solve all sorts of problems.
Exactly. What are some of the most exciting areas of research right now?
Well, one area that's really promising is using Aspergillus oryzae to develop new pharmaceuticals.
Really? Pharmaceuticals? Interesting.
It's enzymes. They could be used to synthesize those complex molecules that form the basis of, many drugs.
Wow.
And this could lead to more efficient ways of producing medicines.
More sustainable, too, maybe.
Possibly. Even paving the way for, personalized treatments.
Personalized treatments? What do you mean?
Treatments tailored to, a person's specific genetic makeup.
Personalized medicine based on enzymes from a mold?
It's amazing, right? It sounds like science fiction.
It does. Are there any other areas where Aspergillus oryzae is making a difference?
Oh, absolutely. Sustainable agriculture is another exciting field.
Sustainable agriculture, okay.
Remember that ability to break down plant matter?
Yeah.
Well, we can use that to create biofertilizers.
Biofertilizers? What are those?
They enrich the soil, promote plant growth.
Interesting. And they're better for the environment.
Exactly. We can reduce the need for those synthetic fertilizers.
Right, the ones that can harm the environment.
Exactly.
So Aspergillus oryzae could help us move toward a more sustainable way of farming.
It has that potential. It's exciting, isn't it?
It is. But with all this talk about, innovation and progress, I also wonder about the ethical side of things.
Ethical considerations. Good point.
Are there any concerns, about risks or maybe unintended consequences of using Aspergillus oryzae this way?
That's a really important question. Like with any powerful technology, we have to proceed carefully, responsibly.
Right. What kind of concerns are there?
One concern is genetically modified Aspergillus oryzae strains.
Okay, genetically modified, yeah.
What if they escape into the environment?
Right, and disrupt ecosystems.
Exactly. Another concern is, the potential misuse.
Misuse.
Yeah, could these modified strains be used in ways that harm human health?
Or compromise food safety.
Exactly. Those are all important things to consider. So rigorous testing, careful regulation,
right,
and open discussions. Those are all essential.
To ensure that we're using this technology safely and ethically.
Exactly. It's about finding that balance
between
harnessing the power of Aspergillus oryzae
and making sure it's used in a way that benefits everyone.
It's a delicate balance.
It is. Well, that brings us to the end of our deep dive into Aspergillus oryzae.
The end of our mold adventure.
I know. We've covered so much.
We have.
We've explored its journey from a wild mold to a, culinary hero and beyond.
It's amazing how much this mold has influenced human history.
It is. And who knows what the future holds for Aspergillus oryzae.
It's exciting to think about.
Well, thank you for joining us on this adventure.
Thanks for having me. It's been a pleasure talking mold with you.
The next time you're enjoying some sushi or steak, or maybe even using an eco-friendly detergent, take a moment to think about that incredible partnership
between humans and microbes
that made it all possible.